Alright guys, so we're going to go over the activity, how DNA controls the workings of the cell. Here we go. Below are two partial sequences of DNA bases shown for only one strand of DNA. Sequence 1 is from a human and sequence 2 is from a cow. In both humans and cows, this sequence is part of a set of instructions for controlling a bodily function. In this case, the sequence contains the gene to make the protein insulin. Insulin is necessary for the uptake of sugar from the blood. Without insulin, a person cannot use digested sugars the same way others can, and they have a disease called diabetes. So here we have instructions. Using the DNA sequence, which is right here, make a complementary RNA strand from both the human and the cow. Write the RNA directly below the DNA strand. Remember to substitute U's for T's in RNA. So sequence 1 is the human strand. So RNA, we're going to have G, C will go to G, A will go to U, T will go to A will go to U, G will go to C, C will go to G, A U, C G, G C, T A, T A, A U, C G, A U, A U, C G, G C, T A, G C, A U, A U, G C, G C, T A, A U, A U. All right, so the next thing it wants us to do is use the codon table in your book, if this is the one in your bell work, to determine what amino acids are assembled to make the insulin protein in both the cow and the human. Write your amino acid chain directly below the RNA sequence. So remember, we're going to divide it into threes. 1, 2, 3, that is a codon. 1, 2, 3, codon. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, and 1, 2, 3. Now remember, we read it G, G, U, so we will go G, G, U, and that's going to be glycine. A, U, C, A, U, C, that's I, L, E, G, U, G, G, U, G, valine, C, A, glutamine, U, G, U, G, U, cysteine, U, G, C, cysteine. Remember, you can use different codes and still get the same amino acid. So we have ACU, threonine, UCC, serine, AUU, ILE. All right, so ILE is isoleucine. All right, so now you're going to do the same thing for the cow. You're going to, G, G, C, A, U, C, G, U, A, C, A, U, G, U, U, G, C, G, C, U, U, C, C, G, L, G. All right, and then we're going to divide it into codons. And we're going to rate it the same. So GGC, GGC, still got glycine. AUC, AUC, isoleucine, GUA, valine, CAA, glucine, UGU, cysteine, UGC, cysteine, GCU, alanine, UCC, serine, UG, valine. So now we're comparing the human gene to the cow gene. How many of the codons are exactly the same? So we have GGC. Nope, not the same. AUC is the same, so that's 1. GUA, not the same. CAA, that's the same. 2. UGU, same. UGC, same. ACU and GCU, not the same. UCC, same. AUU, GUG, not the same. So we have 5 that are the same. How many of the amino acids in the sequence are exactly the same? So it means in the same spot. So 1, 2, not the same. 3, 4, 5, 6, 7, not the same. 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. So 22. Could 2 humans or 2 cows have some differences in their DNA sequences for insulin, yet still make the exact same insulin proteins? Yes, that's because, as you see, you can have multiple codons that code for the same amino acid. So for example, proline has 4 different codons that code for the same thing. So just because one letter is missed doesn't always necessarily mean that there will be a different amino acid. And then find all the codons that can code for the amino acid leucine and list them. So we're going to look for leucine, which is right here. We have leucine right here, here. So we have it in 2 spots. So to get leucine, we can have CUU, we have CUC, we have CUA, we can have CUG. All right, we can have UUA and UUG. All of those code for the same amino acid. So, number 5, diabetes is a disease characterized by the inability to break down sugars. Often a person with diabetes has a defective DNA sequence that codes for the making of the insulin protein. Suppose a person has a mutation in their DNA and the first triplet for the insulin gene reads TAT. The normal gene reads TAG. What amino acid does the mutant DNA and the normal DNA code for, and will the person with this mutation be diabetic? So we have TAT and we have TAG. So this is the original, this is the mutation. So we have, oh, I got that wrong. So this is normal, this is mutation. So TAG or TAT is going to be AUA, which codes for AUA isoleucine. And then this one is going to be AUC, which codes for AUC. This is also isoleucine. Nope. They can do the same thing for this one. Remember, you have to take it, you have to transcribe it, and then translate it. Now we're going to do number 7. DNA sequences can vary. So we're going to do all of this. Which 2 organisms are the most closely related? So we're going to compare the different ones to see how many they have in common. And then you're going to go ahead and do this right here and match it to see which one of these is. And that is it.